Namaskar. Today we will study the difference between nucleotide, nucleoside and nucleic acid. So nucleic acid carry the genetic information and it is a biopolymer of nucleotides. And we can have two types of nucleic acid, ribonucleic acid RNA and deoxyribonucleic acid DNA. So nucleic acid is a biopolymer of nucleotides, and nucleotides is a combination of nucleoside and phosphate. And what is nucleoside? Nucleoside is a combination of sugar and one nitrogenous base. So we can say nucleotide as sugar plus nitrogenous base plus phosphate, and the sugar can be either ribose sugar or deoxyribose sugar. If it is ribose sugar, it is RNA, and if it is deoxyribose sugar, it is DNA. Then the nitrogenous base can be of two types, purines and pyrimidines. The purine is having two rings of carbon and nitrogen, and pyridine is having one ring of carbon and nitrogen. So this is the structure of purine. Numbering we can do it from here 1, 2, 3, 4, 5, 6, 7, 8, 9. So at 1 position, 3 position, 7 position and 9 position we have nitrogen. Similarly, we can have pyrimidine at 1 and 3 position there is nitrogen. So the nitrogenous base which are purine analogues are adenine and guanine, and similarly the pyrimidine nitrogenous bases are three: cytosine, uracil and thymine. So out of them, uracil is present in RNA and thymine is present in DNA. So adenine, guanine, these two purine bases, and cytosine, the pyrimidine base, these three are present both in RNA as well as DNA, but thymine is only present in DNA and uracil is only present in RNA. Now, the sugar, if you see the sugar, so the numbering, this is a pentose sugar, and here its numbering starts with 1 dash, 2 dash, 3 dash, 4 dash and 5 dash. So the ribose sugar has this hydroxy at the 2 dash position, and this hydroxy is replaced by hydrogen in deoxyribose sugar. So based on the sugar we can have the nucleic acid. So ribonucleic acid, the sugar is ribose, and deoxyribonucleic acid, the sugar is deoxyribose. Let us see one example. As we discussed that the nucleotide is a combination of sugar, nitrogenous base and phosphate. So let us see here. In this case, this pentose sugar, this ribose sugar, and the base is adenine, and you can see here the adenine on the 9th position there is hydrogen, and this 9th N9 is now linked to this pentose sugar. So now this sugar and this base forms the nucleoside, that is adenosine. Then it can be combined with the phosphate, and at this 5 dash position, this is the 5 dash position, and on this position the phosphate is attached. If one phosphate is attached, it will be monophosphate, so it could have been adenosine monophosphate, 2 phosphates adenosine diphosphate, and here in the structure we have 3 phosphates, so triphosphate. So adenosine triphosphate is the nucleotide. Similarly, we can have adenosine monophosphate, adenosine diphosphate. So AMP, ADP are also the nucleotides. And let us see here in this example, deoxyribose sugar is there, deoxypentose, deoxyribose sugar is there, and same adenine is the base, and one phosphate is attached. So now we have this sugar and base is the nucleoside, that is deoxyadenosine, then one phosphate is attached, so deoxyadenosine monophosphate, or simply you can write small d AMP, or deoxyadenilic acid or deoxyadenylate, all are same. So if you see the difference between the nucleotide and nucleoside, this base and sugar together is nucleoside, and when the phosphate is attached it becomes nucleotide. Here the base and the sugar nucleoside, then phosphates attached, then it becomes nucleotide. So let us see some examples and how they are named. Suppose the base is adenine, and when the adenine is combined with the ribose sugar, it forms adenosine, and the nucleotide is adenosine monophosphate. Similarly, we can have adenosine diphosphate or adenosine triphosphate. Here I have given the monophosphate examples, and it is the nucleic acid is RNA. Similarly, if the sugar is deoxyribose and the base is adenine, the nucleoside will be deoxyadenosine, and the nucleotide will be with one phosphate, deoxyadenosine monophosphate. So it is a part of the nucleic acid DNA. Similarly, for guanine also the same type of nomenclature. For ribose, guanosine, and deoxyribose, deoxyguanosine, and accordingly the nucleotide, the monophosphates, and they are the part of the corresponding nucleic acids. But the nomenclature differs for the pyrimidine bases. For example, cytosine. Cytosine is the base. When it is combined with the ribose sugar, it becomes cytidine, the nucleoside, and one phosphate attached, it becomes cytidine monophosphate. So it is a part of RNA. But if the sugar is deoxyribose, the nucleoside becomes deoxycytidine, and with one phosphate it becomes deoxycytidine monophosphate. So it is a part of DNA. Similarly, thymine becomes thymidine. So base is thymine, nucleoside is thymidine. So this nomenclature is like this: adenine, nucleoside is adenosine; guanine, nucleoside is guanosine; cytosine, nucleoside is cytidine; thymine, nucleoside is thymidine, and if it is the sugar is deoxyribose, it is deoxythymidine, and with a phosphate it becomes the nucleotide, thymidine monophosphate, which is a part of RNA, deoxythymidine monophosphate, it is a part of DNA. And as you know, uracil is the base for RNA, so it can attach to the ribose sugar and it will form the nucleoside uridine. With a phosphate it becomes uridine monophosphate, the nucleotide, which is a part of the RNA. So this is all about the difference between nucleoside, nucleotide, and nucleic acid. So first we came to know that there are bases, they can be purine base or pyrimidine base, and when this base is attached to a sugar it becomes nucleoside, and when this nucleoside becomes attached to a phosphate it becomes nucleotide. So with one phosphate it is monophosphate, and with two phosphate it is diphosphate, with three phosphates it is triphosphate. Thank you.